Good afternoon, and welcome to this webinar from the Association of Physicians of Great Britain and Ireland. This is on the pathophysiology of mental disorder. I'm Jonathan Kavanagh, Professor of Psychiatry at Glasgow. I'm joined by two colleagues, Professor Basu, also from Glasgow, and Professor Danny Smith from Edinburgh. As I mentioned in the blurb, as it were, mental disorder is very much in the headlines and a focus of many funders at the moment, but it's still exacting an enormous toll on both the health and well-being of people, and the physiology remains stubbornly enigmatic. We hope to show some research approaches that are trying to tackle mechanism and reach across different erstwhile separate medical disciplines. So to that end, we hope to share that with you this afternoon. If you can have any questions put into the Q&A function on this platform. Note that Danny Smith's talk is not live, it's recorded, so any questions you do have for him, can you put them in the Q&A and we'll forward them to him and endeavour to get those answers to you. So without further ado, it's my great pleasure to welcome my colleague, Professor Neil Basu, who's Professor of Musculoskeletal Medicine here in Glasgow, who's going to discuss mapping the brain in order to understand fatigue in systemic inflammatory disease. Neil.
Thank you, Jonathan. And thank you for this invite. I suspect many of you will be wondering why it is that you have a rheumatologist speaking to you for a mental health symposium, not least speaking to you about fatigue. But fatigue is a critical domain of all mental disorders, and it's also a critical domain of the challenges that my patients with inflammatory rheumatic diseases face, and certainly what they share with me in my clinics. So before doing this presentation, I had to re-educate myself on how to diagnose depression. And I believe this is the latest diagnostic criteria which my psychiatric colleagues use. And within that, a key criterion is fatigue. So fatigue is really important here. And I've been vexed, intrigued by this symptom for a number of years and tried to understand it a lot more, particularly because it's so often ignored by our biomedical community. When I'm asked to give talks on fatigue, the audience is often very surprised when I share this data. This is from a general population cohort in the south of England, an analysis I did many, many years ago, where I was interested to know about different risk factors of premature mortality. And of course, I wasn't surprised when smoking came out as a clear and independent risk factor for premature mortality. But perhaps a bit more surprising was the fact that high levels of fatigue came out, despite adjusting for the usual lifestyle factors such as exercise and diet and comorbidity. And this finding has been replicated across the world. So fatigue is really important and worth investigating. Back to my clinical populations, this is simple prevalence data from many studies in rheumatoid arthritis, which have sought to quantify the prevalence of clinically important fatigue in this clinical population. And you can see that across these various studies, it's pretty convincing to say that the vast majority of patients with rheumatoid arthritis have significant fatigue. And further epidemiological studies go on to strongly evidence that this fatigue is one of the principal determinants of impaired quality of life. It's one of the principal determinants of work disability. It can be the difference between happiness at home and divorce. So it has major impact for my patients. And this is data from rheumatoid arthritis. But again, I can replicate this across the entire inflammatory disease spectrum, whether it's lupus or ankylosing spondylitis. And we've all made the assumption that this excess amount of fatigue that these patients experience has something to do with inflammation. So back at the beginning, when my curiosity was first sparked, I, like many of my colleagues and my patients, focused in on potential peripheral causes of this fatigue when trying to understand the mechanisms. And given that rheumatological disorders are, by definition, musculoskeletal in origin, one of the first experiments that I conducted amongst lupus patients was applying multimodal MRI of skeletal muscle to try and understand whether there were any correlates of fatigue severity. And we looked at a number of different metrics, from microscopic structural changes in the muscle to actually getting the patients to exercise while they're in the MRI scanner. You see an image here of one of the cycles that we used. And the purpose of this was to get the juices in the muscles going. And we were getting metabolic readouts through spectroscopy with particular hypotheses around dysfunctional ATP metabolism. But despite these very granular readouts, we weren't able to identify any significant associations with fatigue. So then I parked the inspection of the periphery when it came to fatigue mechanisms and looked more centrally. And this made a lot of sense, given that a lot of the epidemiological studies, again, which look at associations, always seem to find more central associations with levels of fatigue. Things like cognitive symptoms and depression and other mental disorders all seem to correlate most strongly with fatigue compared to peripheral readouts that we have both clinically and experimentally. And I've always had this hypothesis that in my patients, at least, the peripheral inflammation was generating a burden on the brain, which subsequently led and leading to the generation of fatigue in my patients. And if you're not persuaded by that hypothesis, I would ask you to consider the last time you had an infection, another example of an inflammatory hit, I suppose. And I bet you experienced at least a couple of these symptoms that we refer to as sickness behaviors over those few days that you struggled with your infection. And first and foremost is fatigue, but there's other symptoms such as sleepiness, emotional vulnerability, lots of central nervous system generated symptoms which come together, cluster together in response to this inflammatory hit. And I say fatigue is first and foremost one of these experiences that we've all had. So experimenting on investigating the brain in human beings isn't that easy. It's not very ethical to be biopsying our patients' brains to understand the pathophysiology. So of course we rely on surrogate markers. And in particular, I've been exploiting and employing multimodal MR techniques, which go above and beyond the conventional readouts that we're all familiar with as clinicians. Every day there's new sequences coming out, which allow us to understand different dimensions of the brain, often at a microscopic level. So for example, we can get a greater sense of structural abnormalities. We can quantify the volumes of different regions of the brain. We can measure cortical thickness. We can quantify surface area. We can get a whole host of structural metrics. We can also look at the white matter with greater scrutiny as well. There are sequences called diffusion tensor imaging, which allow us to look at the white matter bundles and how they connect across the brain, generating beautiful pictures, as you can see here. And then finally, functional MRI, functional connectivity in particular, does enable us to look at the state of fatigue and how different regions of the brain are communicating with each other in the context of symptoms of interest. So I'll start with a wee bit more inspection on functional MRI. And just to say that it exploits the fact that the paramagnetic strengths of hemoglobin versus deoxyhemoglobin are different. And this difference can be measured by the MRI scanner to produce a signal called the bold signal, which is our surrogate marker of brain activity. And so this bold signal is what you see in this brain here in sort of orange and red. And this is just a cartoon of what's going on in the bold signal with a patient who's just in a scanner doing nothing at all. And you can see how active the brain is, unsurprisingly, even when one is at rest. And this, as I say, is a surrogate marker of brain activity that we can analyze. And there's many different ways of analyzing this data. And I've alluded to the concept of analyzing from a functional connectivity perspective, because symptoms like fatigue aren't going to have one single region of the brain which are pathological. It's going to be communication between multiple different regions of the brain. So what we do in functional connectivity is we look at the bold signal over time across different regions of the brain. And we look to see if different regions maybe are having the same bold intensity over time. And from that, we make the assumption that these two regions, if they are correlating, then they might be communicating with each other. So what we have here is the insula and the posterior cingulate and the time series of their bold signal. And you can see there is an association there. And therefore, from that, we do wonder that in the context of the symptom of interest, in this case, it was pain, that there is communication between these two regions. So we applied these functional connectivity analytical approaches to groups of patients with rheumatoid arthritis, all with varying levels of inflammation. And we were interested to see how the inflammation in the periphery mapped onto the brain. And in the context of functional connectivity, that allowed us to pick up potential inflammatory hubs of the brain. And we did that, and we found that the left IPL and the prefrontal areas were particularly strongly related to levels of inflammation as measured by simple ESR. And we replicated these findings as well. But of greatest relevance was that when we extracted the connectivity indices and related them back to the patient, what the patient was telling us, these regions also related to a number of key patient priority symptoms, including fatigue. And in particular around fatigue, the dorsal attention network and its communication with the frontal pole was really important. We've also looked at fatigue from a structural perspective. This is again from a study looking at rheumatoid arthritis patients, looking at different regions of the brain and their volumes, and relating that to levels of fatigue as reported by the patients. And consistently, we found that putamen in the subcortical regions seems to be larger in patients who have the highest levels of fatigue. Again, this has been replicated. And this is not specific to just rheumatoid arthritis. Other colleagues across the world have identified the dorsal attention network, have identified the putamen and its volume to be significantly associated with fatigue in other clinical populations. So this tells me that what we're mapping here in the brain in the context of fatigue is likely to be more generic than disease-specific. I've got no doubts that there are going to be disease-specific elements, but from what we're seeing so far from these sorts of imaging studies, there's more similarities across different clinical populations in the context of fatigue than differences. But of course, these studies are all cross-sectional, and they can't tell us anything about causality. And it's hard to get towards causality in human experimentation, but there are things that we can do. And this is an example of experiments we're doing in Glasgow at the moment, where based on the information that we're gleaning from our MRI studies, we're now targeting these regions with different non-invasive techniques. And this is a video here of something called transcranial magnetic stimulation, where this is an example where we're actually targeting the motor area, particularly the area which controls the function of the right hand. And when I play the video, and if you have a look at the right hand, you'll see how the magnetic pulses are doing their job in a very focal manner. Let me just play this. So I hope I've shown you that this sort of technology can help us probe the brain. And now we're using this technology to probe these regions of the brain which seem associated with fatigue. And measuring whether the fatigue changes or not, or whether other immune parameters change or not in response to this. And we're scanning patients pre and post, and of course we have shams involved. And this is to help us understand more mechanistically what's going on here with fatigue, but potentially in the future, this could be another way of treating this vexing symptom, for which in truth we have very few treatments for. One treatment that we are studying in the field of rheumatology is vagal nerve stimulation, non-invasive vagal nerve stimulation. This young lady has one of these devices which stimulates an accessory nerve of the vagus nerve just in front of the tragus in the ear. And in rheumatology, there's studies now coming out suggesting that by stimulating the vagus nerve, we can control some peripheral inflammatory pathways. And indeed, this data here showed that disease activity in rheumatoid arthritis in one study significantly reduced following treatment with vagal nerve stimulation. But what we've been doing in Glasgow is observing that these sorts of treatments in these patients also reduce fatigue. And we've just finished a brain imaging study where we scan patients pre and post vagal nerve stimulation. And we've just found that this dorsal attention network is modulated by these sorts of interventions. We're also using imaging, not just to understand mechanism, but potentially as a putative biomarker for treatment response in the future. I did mention previously that we do have very few interventions for fatigue, but non-pharmacological interventions such as exercise and cognitive behavioral therapy are pretty tried and tested and established. So I've recently completed a trial where we randomize inflammatory rheumatic disease patients to receive either exercise, a program which we call PEP, or a talking therapy, a program which we called CBA, and our control arm was our simple education leaflet, which unfortunately is standard care in most rheumatology clinics in the UK. And these are the headline data. We showed that both interventions at our primary endpoint of 12 months was both statistically and clinically important. But you can also see that there was variation in the outcomes, i.e. not all patients responded the same, as is always the case with these sorts of interventions. So what we did do for about 90 patients who underwent this trial is we scanned their brains at baseline. And this is some preliminary data showing that there's some connections in the brain which can predict treatment response. Some of these connections predict treatment response regardless of the intervention, where others are more specific to the exercise intervention or the talking intervention. And it's just like a taster of the potential of these sorts of brain imaging techniques, which one day could be applied in clinical practice. So that was a quick whistle-stop tour of some preliminary data explorations of trying to understand what's going on with fatigue, a symptom which is relevant not just to my rheumatology patients, but to patients with mental health and other long-term conditions. Thank you very much for listening.
Thank you very much, Neil. We'll move on, I think, as I say, and just for the risk of repetition, if you've got any burning questions, can you put them in the Q&A, and we'll endeavour to answer those at the end of the meeting. So next up is Professor Danny Smith, who is from the University of Edinburgh, who's going to discuss aspects of chronobiology.
Good afternoon, everyone, and thank you very much for the invitation to speak to you today. First of all, let me share my screen. So the title of my talk is Bipolar Disorder as a Primary Disorder of Circadian Function. And I thought, by way of introduction, I could share with you some key facts. The first, of course, is that all of biology is to some extent circadian, and also the healthy circadian rhythms are fundamental for health and survival in humans. We also know that our modern way of living is very much ill-suited to the way that our systems for circadian homeostasis have evolved over many millions of years. And then thinking about mental illness, disrupted sleep and circadian function is both a symptom and a cause. So we know this is true for bipolar disorder particularly, where disturbed sleep and overactivity is a symptom of mania, but equally sleep deprivation can cause episodes of mania. Another key point is that many medicines actually work differently at different times of the day, and this isn't really a fact that we've appreciated, I think, in most of medicine, but also particularly in psychiatry. So there's a chronotherapeutic aspect to the way that we use medicines. And then the key point from my talk really is that circadian disruption is the core problem in bipolar disorder, but because of this, it's also potentially a new solution for new treatment approaches for bipolar disorder that really focus on stabilizing circadian rhythmicity. Bipolar disorder, as many of you know, is relatively common. It affects at least 1 in 50 people, although it is very clinically and etiologically heterogeneous. It carries a massive toll for individuals and families, including premature mortality of up to 10 to 15 years and very, very high rates of suicide compared to the general population. It's obviously very costly for the economy and for society and for individuals and families, but we do have a gold standard treatment, which I'm going to come back to in this talk, namely lithium therapy. So what do we mean by circadian rhythms? Well, at different times of the day. So of relevance to this talk is the fact that as we move from afternoon into evening, our melatonin secretion in response to that change in light begins to peak around 9 p.m. and then continue to get us to sleep really as we approach midnight. And then equally other physiological functions are optimized at other times of the day. For example, our lowest body temperature is around 4 in the morning, and our fastest reaction times in the afternoon, etc. So circadian rhythmicity is really fundamental to our physiology. And a key way in which we regulate these rhythms and stabilize them on a approximately 24-hour cycle is light, and particularly light in the morning. So the key Zeitgeber or timekeeper is light exposure in the morning, which really programs our brains to have a circadian function in lots of different ways, but particularly in terms of the timing of our sleep in the early evenings, in the late evenings rather. So light is a key factor here, and I'm going to come back to that as well. In fact, I'm going to try and connect 3 aspects of bipolar disorder, namely circadian rhythmicity, light exposure, and timing of light exposure, and lithium therapy. And my conclusion really is that there's probably a link between lithium, light, and circadian rhythmicity in terms of the way that it is effective for bipolar disorder. I've been part of the International Society of Bipolar Disorders Chronotherapy and Chronobiology working group, and recently we published this position paper, which is really a very in-depth review of the importance of circadian systems for bipolar disorder. And as you can see, this operates at many different levels, from the genetics right through to neuronal signaling, to clock systems around metabolism and inflammation and reward, and right up to rhythmic behaviors like sleep and mood and activity. Perhaps a more digestible way to think about that is these different multi-level organization of circadian disruption and bipolar disorder. So we know that there's been a lot of research recently on identifying genetic variation in genes that predispose people to bipolar disorder. At the next level up, we have research that looks at signal transduction, monoamine transmission, cortisol metabolism, etc. And then above that, we have research that is probably less advanced, but which we need more of, around clock control systems in terms of the autonomic system, metabolism, inflammation, and reward. And of course, all of this feeds in to the symptoms and signs of bipolar disorder. So reduced need for sleep during mania, too much sleep during depression, cognitive changes, suicidality, etc. And then alongside all of that, of course, is the importance of these Zeitgebers or ways in which we regulate these rhythms, mostly light, but also feeding schedules, time of day for meals, activity during the day, social interaction, and of course, stress. So that we really need to move towards research in mental health generally that is ambitious and that takes account of these multiple levels. And this is particularly true, I think, for bipolar disorder. So we want to integrate research on circadian rhythms with research on genetic variation, research on signal transduction, etc. A key point is light exposure, as I mentioned, and this is just to show you how the amount of light that we get in the UK, and this is for London particularly, really does change dramatically throughout the year. So there are massive seasonal variations in photoperiod, namely the amount of daylight that we're exposed to. Obviously, it peaks in the summer months and then reduces rapidly during the winter months. And a key point here is that the rate of change of solar insolation or radiation on the Earth's surface in the northern hemisphere, it changes rapidly around as we move from winter into spring and as we move from summer into autumn. So these are key periods for people with bipolar disorder, for example. We know that, and we've known this for a long time, and indeed this is a systematic review of all the data of seasonal peaks and admissions for bipolar disorder. And the key point is that in spring and summer, we see really a massive peak of manic episodes in bipolar disorder and hospitalizations related to that. And then conversely, as we move from autumn into winter, when there's less daylight, we see a peak of admission for depressive episodes in bipolar disorder. So this is really important and something we've known for a long time, but probably underappreciated both in clinical practice and also in research settings, the fact that there's these quite significant seasonal variations in bipolar disorder, and we need to understand more fully exactly what's driving those changes. It's long been said that there aren't any proper animal models for psychiatric disorders, and to some extent that is true, but for bipolar disorder, and particularly for mania, we do now have multiple systems in which to test some of these ideas around seasonal light exposure, etc. So we have sleep deprivation stressful life events to cause episodes of depression and mania. But actually, given those seasonal variations in depression and mania, we need to think about the importance of the rate of change in solar insolation around those times when we move between winter and summer, and then from summer back into winter. So that we're starting now to think about how to integrate seasonal change in light exposure with animal models for bipolar disorder. And we also now know, and indeed we've known for quite a long time, that individuals with bipolar disorder are super sensitive to the adverse effects of light. This is one experiment from quite a few years ago now, but essentially all of us, when we're exposed to light at night, will experience a suppression of melatonin. So bright light at night blocks melatonin secretion to some extent in everyone. So in this experiment, what these investigators have done is to compare people with bipolar disorder versus people without bipolar disorder in terms of the sensitivity of melatonin under secretion caused by bright light exposure. And as you can see, under normal circumstances, sorry, I'll just go back. Under normal circumstances with no light exposure, melatonin secretion increases both for controls and bipolar disorder, and these differences here are not significant. But when we shine light at midnight, and in this case about 1 hour of bright white light exposed to people at midnight, what we see is that there's a decrease in the amount of melatonin secreted, and in fact this decrease is greater, the suppression is greater for people with bipolar disorder compared to people without bipolar disorder, and that is the case across multiple different light intensities. More recently, a more sophisticated, more detailed approach to this has been conducted by Ritter and colleagues, and they used a protocol over 3 nights, and they used narrow bandwidth blue light to demonstrate that individuals with bipolar disorder are super sensitive to this blue light compared to people without bipolar disorder. So they experience much more phase delay. So the onset of their melatonin secretion is pushed back into the evening when they are exposed to blue light at night. So as you can see here, that is a difference between the bipolar group and the non-bipolar group. So really just highlighting that people with bipolar are super sensitive to the adverse destabilizing effects of light, particularly in nighttime. What about lithium? Well, this experiment gave lithium to healthy volunteers, i.e. people without mental illness, and demonstrated really that lithium could reverse the adverse effect of light on nocturnal melatonin secretion. So that's extremely interesting from the point of view of lithium's mechanism in bipolar disorder, because it raises the possibility that in people who do well on lithium, in people with bipolar who do well on lithium, the lithium may actually be working because it makes them less sensitive to the adverse effects of light in the evening, and therefore makes them more likely to be stable during these periods of rapid change in light exposure as we go through the seasons. So I've mentioned lithium, and I want to take you back over 70 years to its discovery. John Cade, working in Melbourne, first described the effect of lithium on 10 manic patients in the Royal Melbourne Hospital. And at the time when he published this, he said the sedative but non-hypnotic effect of lithium in mania is so specific that it leads to speculation as to the etiological significance of a deficiency of lithium in the body. Now, of course, we know that bipolar isn't caused by lithium deficiency, but it's really lithium is so effective in bipolar disorder. It's such an important thing to better understand so that we can develop new and more targeted treatments. And over the years, there have been many researchers who've investigated the effects of lithium. And in fact, there are many well-described actions of lithium in the body, ranging from monoaminergic transmission changes, second messenger systems, neuroplasticity, transcription factor, promotion, inhibition of GSK3, etc., etc. But more recently, and I think from my point of view, much more interesting are the bottom two of these factors. So the lithium actually shortens the circadian period, and this applies in plants, in animals, and in humans, and also stabilizes rhythms in humans. And taking this a little step further, lithium may actually may have this stabilizing function because it decreases the retinal supersensitivity to light that people with bipolar disorder have. There's just to show you some evidence in support of that, this is data from a clinical trial which identified lithium responders on the left and lithium non-responders on the right, and looked at the circadian aspects of fibroblasts from those two different patient groups, and really just demonstrating that in the lithium responder group, lithium shortened the circadian phase I think to a greater appreciation of the potential for chronotherapies in bipolar disorder. So this is kind of moving away from the predominant paradigm of the last 30 years, where I think psychopharmacology has actually not been desperately helpful in many ways in terms of developing new treatment approaches for bipolar disorder. Lithium, after 70 years, remains by far and away the most effective treatment, and we still don't really understand exactly how it works. So I think the answer to better treatment for bipolar disorders lies in integrating the knowledge about lithium, investigating lithium therapy, but also thinking about how it interacts with light and how that can help us produce better chronotherapies for bipolar disorder. So a nice example of this is a small study of mania conducted by Henriksen et al. And essentially what was tried here, this is in an inpatient setting on about 20 patients with mania who were given blue light blocking glasses to wear in the evening. So logistically a very challenging study to do, but I think really important and interesting is the fact that individuals who were able to keep their blue light blocking glasses on, and so therefore protect themselves from the adverse effects of evening light, tended to get better from their manic episode quicker than those individuals who did not have blue light blocking glasses. So I think that's a small study. Obviously, there are all sorts of limitations with it, but it's very interesting that something as simple as blue light blocking glasses could actually be therapeutic in mania. And then the converse of that, of course, is that there are now several randomized controlled trials suggesting the effectiveness of adjunctive light therapy in the morning during episodes of bipolar depression to treat bipolar depressive episodes. So there's now quite a lot of data on this. Again, this is a very simple intervention, but not something that is used very much in the UK. Reasons for that are complex, but it does look like even some very straightforward approaches like regulating the exposure of bright light in the morning can be a helpful approach for bipolar depression. So as part of the International Society for Bipolar Disorders Task Force, this literature on chronotherapies has been reviewed, and several treatment approaches chronotherapeutically do have good evidence for treatment. For example, bright light therapy is effective for depression, as we've heard. Sleep deprivation may also be effective in severe depression. Dark therapy, such as the blue light blocking glasses, is helpful in mania. And then interpersonal social rhythm therapy is important as a good treatment for the prophylaxis of bipolar disorder. But I would add to this list that other areas we need to think about are the timing of medication in bipolar disorder. So traditionally, we give lithium at night because we need to get a 12-hour half-life measure for monitoring the next morning. That's entirely done for the convenience of doctors. It seems very possible to me that giving lithium at night might not be the best, at nighttime may not be the best time in which to give lithium. And we need to think more creatively about the timing of medications such as lithium, and testing whether different times of the day lithium is more or less effective in the longer term. And of course, there's a lot of interest in developing new drugs that act directly on clock mechanisms as a way to regulate the kind of deranged circadian rhythmicity that many people with bipolar disorder experience. So to summarize my talk, I hope I've convinced you that circadian changes in energy, activity, and sleep are really the defining core clinical features of bipolar disorder. I would say energy changes in bipolar disorder are probably more important clinically than asking people about depressive mood or indeed euphoria or manic mood. It's all about energy and indeed brain metabolism. So secondly, people with bipolar disorder are super sensitive to light and to seasonal changes in the amount of light hitting their eyes. This results in more depression in autumn, winter for these individuals, but also much more mania in terms of spring, summer. And this is, as I said, an underappreciated fact that we should think more carefully about in terms of how we deliver services for people with bipolar disorder. Light in the morning is the primary regulator of healthy sleep, and as I've said, may be helpful as a treatment in bipolar depression. And if we block light exposure effectively in the evening, this may also be therapeutic in mania. I think a key point for my talk that I want to suggest is that lithium, we know, works by stabilizing sleep and circadian rhythms, but what we need to establish is whether or not this mechanism occurs at the level of the retina. Is it possible that lithium decreases the retinal sensitivity to evening light and thereby makes bipolar individuals much more resilient to the adverse effects of changes in seasonal light exposure? Just to end, I know this talk has been all about bipolar disorder and psychiatry, but for a more general audience, I think it's important to think about circadian biology more generally in medicine. This is a really influential, I think, perspectives paper from Sideroth et al. In Cell Metabolism from a few years ago. And they really just say that the importance of Therefore, it is now time that circadian factors became an integral core part of translational research. So obviously, I think this is fundamental for psychiatry and for bipolar disorder, but I also think it's fundamental for the rest of medicine to take more account of circadian biology as we develop and test new treatments. I'll stop there. Thank you very much for your attention. I'd like to thank all my collaborators in Edinburgh as well as internationally. And if you're interested in more information about our work, please go to chronopsychiatry.github.io. Thanks again for your attention.
Well, thanks very much, Danny. I'm sorry about the odd glitch there, but I'll move straight on to my own talk now. Just give me 2 seconds while I share my screen. So again, thank you for the organisers for asking me to speak. I'm going to speak about something slightly different. My interest is in the immune-mediated mechanisms that might underpin major depressive disorder, which has been an interest and a research interest of mine for the last 25 years. I still struggle to some extent with trying to understand how this heterogeneous and rather enigmatic disorder remains free of advances in therapeutic interventions over the last 30 years. When I started in psychiatry, SSRIs were the new great thing. And there's been no real advances in medication since then, I would argue. And we still struggle with not only the impact it has on health and well-being, but it's also very significant mortality. My earliest experience actually as an SHO in Edinburgh was of the death of a young woman through suicide who had been treated with an SSRI for the first time, and yet it did not manage to save her life. So my concern and drive has been to try and understand this disorder from a biological mechanistic perspective. We have a problem, however, in the sense that we have a lack of mechanistically specific human biomarkers in psychiatry generally. And in depression specifically. There's thus a paucity of novel, tractable, new molecular targets. And we struggle with poorly predictive animal models. And half of my work is actually based in basic science. So we use a lot of animal models, but none of them are of depression. So today I want to say something about how mechanism leads to molecular targets and targets to drug development. And so I'll focus on one potential biological mechanism in psychiatry, namely immune-mediated disorders. The limitations of human studies to parse mechanisms, which Neil touched on, requires us really to reverse translate. And I'll give lastly an example of experimental medicine clinical study based on such mechanistic science. So this one particular biological mechanism is essentially immune-mediated inflammation, and there are four main arguments around why that might be important in at least a subset of depression. And I mentioned at the beginning how heterogeneous depression is. It's an extremely common disorder, but about 25% of patients exhibit some level of low-grade inflammation. And that is coterminous with their treatment resistance to the currently available monoaminergic antidepressants. But perhaps most convincingly, inflammation itself or inflammatory cytokines can induce depressive symptoms in those people who have otherwise been free of depressive symptomatology. A classical example being interferon therapy for hepatitis C. And in those patients, about 50% of them will develop major depressive disorder de novo. And what's interesting about that is that not only do they develop depression, that depression doesn't go away on the cessation of the interferon therapy. And in fact, it's some form of priming might go on here, where a patient treated with an interferon, an inflammatory cytokine at the end of the day, becomes depressed over time and develops this remitting relapsing disorder. I've touched on the fact that you can see elevated cytokines should you choose to measure them. IL-1 beta, TNF-alpha, and IL-6 are the most commonly described in the peripheral blood of this subset of psychiatric patients, this subset of major depressive disorder. And I've also mentioned how that's coterminous with this resistance to treatment. Lastly, and the most recent data, I suppose, is around the idea that anti-inflammatory therapies, this development, particularly in rheumatology and other medical disorders, of highly targeted anti-cytokine therapy happen to have antidepressant effects. And those studies have almost entirely been done with depression as being a secondary outcome, but nonetheless, and you can see that on the top right, there's significant evidence that there is an improvement in depressive symptomatology in patients who've been treated with drugs like infliximab, for example. But stratifying those patients on the basis of inflammatory markers is key to understanding that. You do not see that improvement in patients who are not stratified on that basis. I've touched on and borrowed again my colleague Neil Basu's slide here, which touches on the relationship between cytokine change in the periphery, or inflammatory change in the periphery, and change in connectivity in the brain, which is also related to and coterminous with structural change. So if we accept that there is a degree of inflammatory involvement in major depressive disorder and may well be a biological culprit, how does the periphery communicate with the brain from this perspective? Well, there are two main routes: humoral routes, where there's activation of the blood-brain barrier endothelium, a biologically active entity in itself; active transport of cytokines across the blood-brain barrier. Interestingly, there is an active transporter protein for TNF, the reason for which is still unclear, other than we know that TNF is important for myelination. There's direct access through circumventricular organs, so the blood-brain barrier doesn't cover the brain in its entirety, and activation of cells within that. The more rapid route is neural, through vagal nerve afferents, and that stimulation of the vagal nerve in the periphery, leading to signaling in the brain much more acutely. These panels at the bottom really touch on the different methodologies that we have for trying to measure this relationship between peripheral immune activation and brain change as a consequence of that. And we'll touch on those towards the end of the talk, but essentially they turn around functional MRI, which Neil's described, MRS, which gives us an increasingly exquisite reference point for brain biochemistry for things like glutamate, which has a hypothetically important role to play here due to its involvement in excitotoxicity, and the more molecular forms of neuroimaging, PET and SPECT, which allow us to use specific ligands, which are increasingly important in understanding inflammation in the context of the brain. But as I said at the beginning, there's a limitation in human studies to parse mechanisms. We're very limited in what we can actually do to people, rightly so. And so therefore we must fall back to some extent on reverse translation and in laboratory models. In our lab, we've been using a model called Aldara, which is a skin-based model, which is actually a model of psoriasis, but it's a TLR7 agonist, and the imiquimod, which is the active component of the cream that you put on the mouse's back, binds to toll-like receptors and results in our hands in an upregulation of cytokines and chemokines within the brain and within the periphery. But that change within the brain is both at the transcriptional level, but also at the protein level, importantly. And this graph here shows a variety of cytokines and chemokines that are significantly upregulated over time in the brain of these mice. And that is interesting from a variety of perspectives. Not only is it upregulated, but it's upregulated in a distinct way from what's happening in the periphery. And in this model, actually, that particular TLR agonist gets into the brain. So it provides us with a very direct form of neuroinflammation, which is almost like a mild encephalitis without the need to inject the brain, which is the typical way of stimulating neuroinflammation. So we don't have to use intracerebral ventricular injections. We can get a mild form of stimulation using this particular model, which we found fortuitously, in fact. So that leads to, as I say, a variety of changes over time in these cytokines and chemokines. This is an example of one chemokine involved in leukocyte recruitment. Sorry, my apologies. Let me just go back again. That chemokine involved in monocyte recruitment into the brain. And that's the other interesting thing about our findings here, that we saw a significant ingress of leukocytes into the brain in the context of this immune stimulation. Those were predominantly monocytes, but they also included T cells and a variety of other natural killer cells. This was lastly related to behavioral change. In this case, this was a very basic but nonetheless sensitive model of mouse well-being, for want of a better expression, where mice will naturally burrow. They'll burrow all sorts of materials, and that is a hedonic behavior in mice. It's a naturally enjoyable thing that rodents will take part in. And what you'll see here is that the treatment group here show very little change, and the weight left in the tube is the amount that's been burrowed. Whereas here is the treatment group, and you can see that oh, sorry, this is the control group here in light grey, which is what you'd expect to see. In other words, you'd see a decrease in the amount of available burrowing material. But in our treatment group, you see that hardly changes. So there is, if you like, an anhedonic response as a result of this immune stimulation. So a quick summary of this section. Brain resident cells can respond directly to significant cytokine and chemokine response, and brain resident cells, of course, being microglia, neurons, oligodendrocytes, and astrocytes. There's evidence that in our hands of an increased expression of both microglial markers and astrocytic markers and changes in cell morphology. We see immune cell trafficking into the brain, but these cells can be found throughout the brain. And when I look down the microscope at these brain sections, there's no obvious localization pattern. They're peppered throughout the brain parenchyma. And that's leading us to a series of other questions around this, because all of this relates to general systemic inflammation, but can we be more molecule specific? The great advances that have been seen in rheumatology, dermatology, and gastroenterology have been really over the last 20 years around these highly targeted molecule specific therapies. So the next stage we want to take part in here is to develop and grow or breed, I should say, a transgenic mouse which will express solely TNF, and this will allow us to switch on soluble TNF expression at relevant time points, and importantly, in specific cell types, to provide both an acute and a chronic expression of inflammation. And using Cree technology, we can control the onset of this overexpression of TNF. We've got a variety of different promoters available to us for microglia, for monocytes and macrophages, and for endothelial cells. And that particular promoter is important because this is for brain-specific endothelial cells, because the endothelial cells themselves are not passive in this process. In fact, they're highly active and play an important role in the brain's neuroinflammatory response. So this will allow us, I think, and I hope, to develop a molecule, inflammatory molecule specific model to test some of these mechanisms. So if that's molecule specific, what about cell specific? I touched on the fact that we're seeing this ingress of immune cells into the brain in the context of a stimulus, but also this upregulation of activity amongst microglia, perhaps not surprisingly, but also amongst astrocytes and neurons themselves. So amongst the immune cells, which are the cells that are important? Is it predominantly monocytic? Is there any role for other non-myeloid cells, for example? Are we seeing any lymphoid cell activity? Where are the key sites of action within the brain? And how do the neural cells there interact with these cells? And so that's the next series of experiments that we want to take prosecute. One is to use, and this is, I think, an important point here within the context of the pathophysiology of mental disorder, is that we really have to embrace advances in different methodologies that are available to us, and the ability to determine and to describe in increasingly exquisite detail both the transcriptome within the brain and the regions of interest, and how they interact with other cells is increasingly important. So technologies such as Codex, which is, if you like, a very elaborate form of ISH, will allow us to identify regions of interest within the brain. Using spatial transcriptomics, we will be able to look at taking those regions of interest, the entire transcriptome, so the entire genome, and look at exactly what's up and down regulated. And that will also inform us what cell types, by dint of their transcriptional signature, are active within that particular region in the brain. And using available databases such as CellPhoneDB, we can assess cell-to-cell communication. Next, we want to look at and identify the neuronal ensembles. So obviously, neurons are key to all of this because we're interested ultimately in behavior. And we would like to identify those neuronal ensembles identified through the Codex technique. And interestingly, there's a new method. It's not particularly new, but there's a method that captures CFOS, which is an immediate early gene expressed by neurons when they're activated. And we can capture those in the immediate moments when they are neuronally active. That will allow us then to determine which groups of neurons are important in this particular stimulus, and conduct electrophysiology experiments to determine their readout using various forms of optogenetic technique. And the last part would really be to determine what's important, what's not important. And this is where a technique such as chemogenetics will come in and allow us to use DREADS, both inhibitory and activating. And that will also help us with understanding the effects of different forms of drug inhibitor, both at the cytokine and chemokine level. So those will help us answer those key questions. What is the whole brain landscape transcriptional profile in the context of neuroinflammation using codex? That will help us identify the regions of interest and then look at the whole genome analysis in that specific region using the state-of-the-art geomics techniques, these forms of spatial transcriptomics. We next would look at connectivity using the FOSS trap technique, this ability to capture the CFOS expression that I mentioned in active neurons. That will allow us then to use optogenetic and EFIS technologies to work out what that connectivity is. And Neil touched on connectivity in some detail in his talk, but looking at this at actually almost a neuron to neuron level is the next important step, which can only really be done in the mouse. And lastly, as I said, to use chemogenetic techniques, which allows us to use these very designer, very specific drugs, which are either excitatory and inhibitory, to then overexpress or underexpress that activity and look at the relevant behavioral readout. I want to just finish by going back into humans with an example of an experimental medicine study based on some of this mechanistic science that we talked about, these molecular targets that have been so effective in rheumatology and other medical disciplines. For example, IL-6 and TNF antagonism. So here, our hypothesis is that looking at anti-cytokine biologics, what is their antidepressant effect and what's the mechanism underpinning them insofar as we can ascertain that in humans. So we're hypothesizing with this current study that anti-TNF therapy will affect changes in brain network connectivity and glutamate quantification. So why am I picking glutamate? Well, I'll say something about that at the end. We're recruiting groups of rheumatoid arthritis patients who show changes in brain network connectivity and glutamate quantification, and that would be correlated with these 6 behaviors that Neil talked about, key of which is depression. And that these patients will also show changes in monocyte infiltration into the brain that's similarly correlated with depression, fatigue, and other sickness behaviors. How on earth are we going to do this? Well, one of our hypotheses is based on the idea that this upregulation of cytokine and chemokine activity that you get with inflammatory disease is key to resulting in sickness behaviors via an upregulation in extrasynaptic glutamate. Why? Well, TNF and other pro-inflammatory cytokines increase glutaminase. In other words, they will increase the enzyme that produces glutamate, and importantly, will decrease the activity of glutamate transporters expressed predominantly in astrocytes. And those transporters are responsible for maintaining glutamate homeostasis in the synapse. So in essence, you get a net increase in extracellular glutamate. And the argument here is that excess extracellular glutamate is neurotoxic. This is an excitotoxic neurotransmitter which damages neurons and also causes chaotic neural firing. Measuring that is increasingly possible using high field strength MRS, and that's the plan there. The monocyte recruitment into the brain that we saw in the mouse is fiendishly difficult to measure in humans. And so as part of this experimental medicine approach, we are using a SPECT approach where we will take the patient's own monocytes, we'll sort for CD14 positive monocytes, we'll radio label them, and then re-inject them into the patient and measure whether or not there is any recruitment into the brain by dint of a signal using the usual SPECT technology. Now that's essentially an all or nothing signal, but this hasn't been done before, so we have to start somewhere. And so the plan is to take rheumatoid arthritis patients, as I say, who are expressing these sickness behaviors, depression, fatigue, and cognitive impairment, and use these neuroimaging technologies before and after TNF inhibition. And that will, I hope, allow us to ascertain the roles of two potentially mechanistically important processes. One is glutamate expression, the other is monocyte recruitment, and whether or not TNF antagonism makes any material difference to that. So I'll stop there and thank my group and my collaborators and funders. Thank you very much for listening. So I think we have one question from Christian Delez. I think that's from Neil. Are there differences in functional and imaging parameters between fatigue of different origin? For example, post-viral versus fatigue associated with chronic inflammatory disease?
Very good question. I said at the beginning of my presentation how a lot of the methodologies are advancing by the week, and that causes challenges when we're trying to compare findings between different studies, because often pipelines from an analysis perspective are different according to investigator preferences. But despite that heterogeneity, there are similarities which are emerging, particularly around frontostriatal connectivity, regardless of the underpinning clinical population, whether it's coexisting with an inflammatory disease or whether, as the question asks, whether it could be post-viral. But we're beginning to learn there may be as many differences, but at the moment, there does seem to be some shared mechanisms.
Okay, thanks, Neil. There are another couple of questions that were forwarded. Is there any relation between polycythemia and mental disorder? The answer to that is probably not. There is a relationship between polycythemia and delirium. So in the stricter sense of the word, it is a mental disorder in the sense that it causes mental symptoms. But the relationship between polycythemia and depression, for example, has never been shown. The only large study that was done was in relation to trauma in, I think, the East Japanese earthquake, where they found patients had high levels of polycythemia, but they showed no relationship to psychological dysfunction there. And this was a huge study of something like 27,000 patients. Next question was, can perfect mental health prevent suicide? I think the answer to that is probably not, for a variety of reasons. One is there isn't really perfect mental health. But the studies that relate mental health to suicide show that any form of mental disorder puts you at a higher risk of suicide. I think the obvious source of that question, I think, is that if you can resolve that, does that necessarily mean that you prevent suicide? Sadly not. I think that the biggest thing you can do is to treat rigorously and effectively the mental disorders that we've been touching on today. Those are the key and biggest explainers of the variance, if you like, and the biggest contributors to this mortality. But it won't be the whole thing, and it won't be the whole story. And sadly, there are other reasons for people taking their own lives. The last question I've got here is mental health related to financial security? Well, yes, in the sense that financial insecurity is a common form of environmental adversity, if you like. And undoubtedly, the role of stress is key to the development and certainly the stimulation, if you like, of mental disorder, in the sense that if you are at genetic risk, for example, of bipolar disorder or depression, then we know from a whole variety of epidemiological, clinical, and indeed biological studies that chronic unpredictable stress is a major etiological factor in stimulating those symptoms. I think we've got another question from Dr. Mahmood. Shall we receive the presentation soft copies in our emails? I'm happy with that if the other presenters are. Yeah. Okay. I don't see any more questions at the moment. I do have, just while we're waiting to determine whether there's any more, there is some housekeeping for the Association of Physicians. Oh, we've got another one here. Dr. Promi, is there any relation between arrhythmia and mental disorder? that's a slightly challenging question. There is undoubtedly a relationship between cardiovascular disease and mental disorder, which is extremely well described. And in fact, cardiologists are well ahead of the curve, really, amongst physicians in linking depression to poor outcome. Arrhythmia itself, I'm not sure if you're meaning just, as it were, an electronic malfunctioning, but as a manifestation of potential cardiovascular disease, then yes. And in fact, it's one of the more worrying aspects of cardiovascular disease is that the development of depression post-acute coronary syndrome puts you at significantly greater risk of death from cardiovascular disease, not from suicide, but from cardiovascular disease. And so that's why I think the cardiologists deserve a lot of support and praise for that, because they've recognized this and realized that monitoring patients for depression is extremely important, given the fact that they are already at a higher risk of death. That was just a thank you note. Right, okay, I'll just put in some of these housekeeping matters. So the Association of Physicians of Great Britain and Ireland will continue to host seminars in this series, Emerging Issues in Medicine, across the autumn. The next webinar is taking place is aimed at early career researchers navigating a career in clinical academia, and is next Thursday from 4 to 6 p.m. and registration is already open. The annual meeting for the Association of Physicians is taking place in Liverpool next year, 2023, from 20th to 21st of April, and the information is available on the website. I've got two other questions here. Is mental health disorder related to a lack of confidence? Well, I think that's the problem with things like lack of confidence is that it's a deeply subjective description and a deeply subjective thing to measure. I think that it might well be argued that the other way around is also the case, that mental disorder can result in people losing confidence socially, can lose confidence in their ability to function in everyday life, and indeed maintain gainful employment. Keith McAdam is asking, in older people, delirium seems to be triggered by urinary infections. The mechanisms behind that, really interesting question. The one idea is that you are essentially inducing an acute inflammatory response with something like infection. So an example in animals would be something like LPS, where you induce this very acute cytokine storm almost in the periphery. What I've tried to say in my talk is that that communicates in a very direct way with the brain, both neurally and humorally, and that delirium is, if you like, a neurological response to that. Now, how the mechanisms happen in terms of how that communicates with the brain, resulting in changes in neural communication, is the kind of thing we're trying to address. What is interesting, though, about delirium, and is something we've been looking at more recently, is that this is something that's seen extremely commonly in medical wards. It's a major source of mortality, but it's also something that puts patients in a significantly greater risk of dementia. And that's something that we weren't aware of until relatively recently. And you'll be aware that disorders like Alzheimer's, but other forms of dementia, are highly inflammatory in their biology. So if you like, this might well be something that triggers and primes the brain in a way that is less than optimal and leads to the devastating disorders like dementia. So I'm not sure that quite answers your question, but it's certainly a matter of communication between periphery and brain, and some of the mechanisms I've touched on might be the culprits there. More questions. Is mental stress causing eyesight problems? I'm a student. I've noticed that at my exam time I feel a problem in my eyesight. Well, I have no idea as the answer to that. I think is stress generally something that leads to eyesight problems? Interesting question. I think that might be something for Professor Smith to answer in terms of the impact on retinal biology. Well, I'm just looking at the organisers. I think we're probably out of time. Any other questions we'd be happy to answer if the organisers can forward those to us. We'll